Okay, so maybe we will move on. We already talked about the urgency to get more data, to establish registries, and to establish clinical endpoints in rare disease. And the next talk is from Maya Hempel. She's talking about our efforts for an European patient registry.
Yeah, and I'm very happy that I can present the work from Sebastian and Verena. It was a hard work for more than 2 years. And finally, we got a registry. This is we built it up like to enter it by a website, our website from the Institute of Human Genetics in Heidelberg provide the enter point of this registry. The families can easily go to this website and find the information and informed consent on this website. They can sign it electronically, and afterwards they will be asked for send us these genetic reports, and if we confirm that there is an DAP50Q constellation, we send them back by email the invitation for the REDCap registry. And then the patient fill out these questionnaires. They provided at first in German, and now we have questionnaires in French and English. And in addition, the parents sent us reports. These reports will be approved by a registry team, and the data will be filled in the database as well. And we have a lot of variables we ask for, and we document it. And for annual follow-up, it's a fewer variables, but nevertheless a lot of them. We have we're very excited to see our enrollment rate. Within 3 months, nearly 100 parents and patients, and we are very grateful for the parents group that have hard work on this. And I will present you some data from this registry we have extracted 1 week ago. The median age is about 8, with the youngest patient 12 months and the oldest 47 months. We have enrolled more males than females. I was very interested in the first symptoms the kids expressed. This was in nearly half of them muscular hypertonia. The youngest age of onset was right after birth. The patient oldest expressed some symptoms was 3 years old. And you can see that unusual behavior is a second first finding in these kids, followed by global developmental delay. We understand that we can have a lot of data from this database and comparison between the EDIC and interstitial duplication. And for instance, in motor development, we see that there is a trend for different developments depending on genotype. But so far, this is not significant, but hopefully we can show that there is a genotype-phenotype correlation, not only between interstitial duplication and IDIC, but probably with the constitution of IDIC as well. We know that the parents are very interested in epilepsy and kind of epilepsy the kids may develop. And what we can say in this moment is that that the patients with interstitial duplication do not develop infantile and juvenile spasms and do not express status epilepticus. That is what our data show. Okay, at the end, this is only the beginning of our journey through this deep phenotyping of the cohort. We are very happy that so many families agreed to take a part of them. Our dream is to establish an international and European international database, or exchange the data to dive more into the phenotype, in the specific phenotype, and in the genotype-phenotype correlation. This will help us to understand the pathophysiological mechanisms, to find endpoints for clinical trials, and to find specific treatment options. Okay, thank you.
Thank you very much. Please keep your questions in mind. We'll have 3 presentations, and after that, a brief discussion. For the next talk, I can introduce Marie-Claire de Witt from Rotterdam, and she's talking about the Dutch patient registry.
Thank you very much. So we have all our information cut into different sections of this program, but this also gives us an opportunity to all stand here. So I'm the pediatric neurologist from our team, and I've been, as Karen said, we've been working together for I think more than 15 years already, for starting with the Angelman group. So it's really helpful that we have this structure in place that the Duch15Qs can roll into, and we can get a sense of what we're looking at. And this is very inspiring to hear all this information here, and I'm impressed with the number already collected in Heidelberg. We're smaller, but we're growing. Let's see. So what I'm going to present right now for these 5 minutes is the structure of our registry, and most of the data that comes out of it will be presented by Karen later. We had the goal of collecting as many patients in the Netherlands to get some information. It already been said why natural history information is so important. It's important to have a standard of care, to get a sense of the full breadth of the spectrum, so you can give better answers to the questions parents have, and also to collect outcome measures for hopefully further studies to improve care. So we wanted to see where the patients are in the Netherlands, but also get some information out so we can get a better sense of the syndrome. So I'll go. This is not so very interesting. I've mentioned the rationale, so I'll go to the study design. We did a similar thing. We had a prospective survey based on online survey that families could enter through a very similar system. We don't use the red cap system, but we have Castor, but it's almost the same thing. And we had a student starting inclusion of these patients in 2024. So these were our own patients that we already knew, and patients that we collected through contacts, through internet, all that sort of thing. And they get a questionnaire with prompts, and both children and adults were included. We wanted to describe the clinical features regarding epilepsy, behavior, child psychiatric diagnosis, and development, and also the genotype information, so we could do genotype-phenotype correlations. Apart from the survey, we asked the Caracol questionnaire and did a CGI, so that's a clinical global impression of functioning that's used a lot in different syndromes with disabilities, intellectual disability. And physician reports are possible, and parent-reported information in other patients. The neurodevelopmental assessments we do in our clinic, or which Dr. Van Hopen, who's in the back, does when she sees the children, are an IQ test, and mainly the Bailey. Here it says the Bailey 3. We're moving on to the Bailey 4 this year. It's been translated. The Siebel, the communication matrix, has been mentioned already. And in children that have a higher level of functioning, another appropriate test is chosen. And then there are also tests done to see if there's an autism spectrum diagnosis or autism features and ADHD. So the survey was based on the latter survey that's done by the international or the US group the ASD15Q Alliance, and it's a very extensive questionnaire, so we trimmed it a little bit so that we heard it takes about an hour to fill in, and the latter takes about 2 or 3 hours, I think, so depending on if you have a lot of things to say. So I think this works for us. We also use this database now for our clinic, but it would be very useful to compare with the Heidelberg, I think, to see if we could combine our data. We would be very interested in having a European database. So the numbers we reached, we have our own patients, 31, and an extra external patients, so those who are not yet visited our clinic or were adults of 11. Even though we tried to contact everybody, one mother or father was not able to fill in the questionnaire, so we included 37 patients in our survey, in our database right now, and Karen will present the results in our next talk. To be continued.
Thank you very much. Joachim Sobel, the next speaker, couldn't come today, so we will directly move on to Italy. And I'm happy to introduce Alessandra Rossi, talking about longitudinal clinical data from Italy.
Okay. Good morning, everyone, and thank you so much to have the opportunity to be here today. I will talk about the phenotypic spectrum in patients with chromosome 15 long-term arm duplication syndrome, and I want to show you the Italian experience. Okay, my disclosure. Okay, so Duch15q syndrome is a very complex syndrome, and we know that patients with isodecentric IDIC syndrome have more severe phenotypes than those with the interstitial duplication. And what about our purpose? Our main goal was to analyze a novel large cohort of patients with this syndrome to deepen the phenotypic understanding. And what about the methods? We collected previous unpublished patients with IDIC and interstitial duplication seen at the Eugenio Media Research Institute in Conegliano in Italy, and recruited through the Italian Duch15 Family Association. And demographic, genetic, electroclinical data were retrospectively reviewed, and long-term follow-up of comorbidities was evaluated. These are our results. I want to underline that at the moment we recruited nearby 60 patients. So this is only a slice of our cake. But at the very beginning we had 20 patients, 9 male with IDIC, with a median age of 11 years old and half, and 3 males with interstitial duplication, with a median age of 12 years old. And what about epilepsy in our initial cohort? 90% of IDIC showed epilepsy, while only 2 patients with interstitial duplication had epilepsy, in particular with age of onset in the first group at 7 years old, in the second group in the 2 patients at 10 and 15 years old. What about epilepsy syndrome? A classification was possible only in half of the first group, in particular 4 with West syndrome, 5 with Lennox-Gastaut syndrome, 1 evolving from West syndrome, and the rest with developmental and epileptic encephalopathy. Regarding the 2 patients with the interstitial duplication, 1 had Lennox-Gastaut syndrome and 1 febrile seizures. And what about the pharmacoresistant epilepsy? The first group, in 72% of cases, had a pharmacoresistant epilepsy, and only one patient with interstitial duplication had pharmacoresistant epilepsy. Let's go on. What about the developmental delay and the cognition? 100% of all the entire cohort had developmental delay. In particular, the first group, in 100% of cases, it was severe, while in the second group, mild in 29%, moderate in another 29%, and severe in 42%. What about, for example, walking abilities? They were independent in 55% of cases in the first group, while they were independent in 100% of cases regarding patients with interstitial duplication. Regarding the psychiatric and behavioral features, only in this case we had this was a very important comorbidity, especially autism spectrum disorder. And only in this cohort, we showed hypotonia in 80% of patients with IDIC, and unfortunately also an early mortality. I think it could be a main goal of future research. In our case, 3 patients, 15%, died, in particular 2 for cancer, 1 for testicular cancer and another one for a lymphoblastic acute leukemia. Maybe there are intracellular patterns that we could study to understand if there is a correlation between this syndrome and an increasing risk of tumors. It could be very interesting and important for the natural history of the syndrome. So in conclusion, we outlined the clinical features in both interstitial and IDIC patient, but this is only a partial analysis of a broader project involving the Italian patient from the Non Solo 15 Family Association. And I like to remember that research is not a one-player activity. And so thank you very much for the Family Association, because I think very often they are the heart and the force for future research. And it's important to create a data set for collaborations with the other research group for prospective studies and natural history studies. And this is in order to identify the most significant unmet needs of this population. Thank you for your kind attention.
Thank you, Alexandra. So now we have time for discussion. It's good because we have some more minutes because of the missing presentation. Are there any questions? And also, please stand up if you ask the questions.
Okay, thanks. Thank you for both of the presentations. It's quite interesting. I think it's one of the things that I noticed, and I also saw you looking, is the difference in like the cancer. I'm lost. Oh, there. Have these patients been sequenced any further to see if they have additional genetic abnormalities?
So basically, we have, I think, 3 or 4 premature deaths out of 60 patients. Most of them happened a few years ago, and we didn't see them directly. So it's all retrospective data. We know that 2 of them had cancer, basically, but they were not tested further. Another thing that, I don't know if you have seen also in this also your experience, some of these children, especially when they are young adults, especially with the severe epilepsy, they often have severe feeding problems, and also I'm aware that there may be different kind of approaches. For example, in Italy, we tend to have very few children with, for example, gastric tubes or assisted kind of feeding, and this may also be a sort of component that may favor also premature death in a sense. Okay, so it's also about the approach you have of caring of the children. This is a decision that you take with the families, basically. But I think it will be interesting to look at the causes of premature mortality beyond SUDEP, which is obviously one of the potential causes.
Do you guys have anyone with cancer? Yeah, we also have one. Without cancer. Okay, we have one child with leukemia, but other than that, in our cohort, there were not, no.
I was not aware about the tumor predispositions. No.
But I also think that testis cancer and AML are also some kind of cancer which are more frequent in children. So maybe it's just a coincidence.
In Italy, do you have some registry or something like this?
Yeah, basically this project is going to be a registry, okay? But we were wondering whether it would be better to do a national registry or basically contribute from the start, for example, to a European registry, because it will be basically a double work, and I'm not sure if it will be basically more kind of effective and more useful for the families to take part to a European registry from the start. So it's something that we are discussing also with the Italian Association.
Yeah, I would say we are open for this because it was a hard time for us. We suffered so much to set up it, and I think you have the same thing in the Netherlands, so you are very well invited, and we can discuss it afterwards. And do you have something like a specialized appointment clinics or outpatient clinics? This is what you have, yeah?
So yes, so basically we also kind of take advantage on the experience with Angelman syndrome, because we are National Reference Center for both syndromes. And we'll basically, yeah, we have a sort of a specialized clinic. Also, we have a bias regarding epilepsy prevalence, because most of the children are referred to us because of epilepsy, because of drug-resistant epilepsy. So I'm not sure we are capturing the actual prevalence of epilepsy. But yes, we have this sort of specialized clinic with child neuropsychiatrists, epileptologists, and all sort of kind of rehabilitation workers, so speech and therapists, kinesiologists, and things like that. So we try to offer a holistic sort of approach to the families.
Thank you.
So I think in all of these registries we have a lot of patient-reported outcomes, but in order to harmonize the data, I think we also have to talk about the more complex data, especially I think the EEG data and and genotype data. So maybe how do you handle it in Rotterdam? You was first.
Yes, that exactly triggered my interest. Well, in Rotterdam, we'll be able to connect to another project that we're doing. We have a small subsidy that we can use to enter the children with Duch15q in our Child Brain Lab program. So we have a function lab. For the last 2 years, we've set it up in the Sophia Children's Hospital for the Pediatric Brain Center. And there we see children for an in-depth phenotyping. So they get a high-density EEG with a wet cap. We do evoked potentials, auditory evoked potentials, and an eye test. We do a test with eye tracking. simple and more complex measurements. You can do the simple ones with just looking at the screen. You don't have to do anything. But then the more complex, then it becomes a game, and you have to do different things. We look at different outcome measures, and we do a gait analysis and a motor functioning analysis, and also an IQ test and a language test. So this is our next step. We would like to enroll the children that we see in this pediatric brain center carousel, and there are also prompts that we ask parents to fill in. We're doing this for different syndromes, and I think, or our goal is to get a really in-depth phenotyping and also be able to do these measurements to see if we can predict outcome later on, if we can detect deviations from the expected progression in their development, so that we may be able to treat the appropriate children, and also have a situation or have a infrastructure in place that should there be a possibility of doing interventional trials, that we can actually measure the results.
So you have more like one center to do the EEG and all the physiological tests, because I think for our registry it's also really a challenge, because the individuals have a lot of EEGs, but maybe these EEGs come from different places, and it's also hard to address this problem.
I think if we were able to have a European network, we could do the natural history data, but maybe also collect some important data and see who is good at the analysis of this. Of course, just basic EEG, you cannot do everything with it. With the high density EEG, we're trying to develop. Not me. I'm not an AI expert, but I have a machine learning expert colleague who's trying to do network analysis. But for basic, like power analysis of different frequency bands, you can use basic EEG, and we could. I mean, there's already something known about DFTQ, but not enough yet. So I think that might be something that we could combine in a project, and that doesn't need to be very expensive, but maybe just a little bit of funding could help there.
Yeah, thank you very much for sharing this information. I would like to say that we are also collecting high-density EEG data. I think that it would be really great to have a sort of European data set where we maybe use standard EEG, and we can sort of open source environment where you share EEG, and you can find a sort of common denominator between, even although they are being done in different centers, it may not be a problem, okay? And then you can have a smaller kind of sample of children studied with more kind of complex, sophisticated examinations, such as high-density EEG, and together they will be able to kind of respond to different research questions, basically. So it will be really important to do that. I have a question for the other colleagues. What do you think about endpoint measures? Do you think that we have the right ones, or there is room for improvement? Because we have a debate also with Verena yesterday.
You can go next. There's always room for improvement. I think one of the goals, of course, as has been said, that they identify the unmet needs, and there are definitely endpoints to be found there. So, but that also we can learn from other syndromes. So the endpoints of cognitive, language development, behavior, epilepsy, prompts are, I think, well established. And then what we also need are intermediate endpoints, like, for example, with the EEG, because a lot of these endpoints are far in the future. And if you want a better outcome in 5 or 10 years, you need to know what you need to do now to reach that endpoint. So any intermediate endpoints or biomarkers would be most welcome. From my perspective, EEG is promising, because also by my something that I know a lot about, but there might be other things. So Karen is also looking much at metabolic markers and things like that. Maybe you can say something about that.
Yep. Oh, I'm going to talk about it later, but in the same project that Maria Claire is addressing, the Child Brain Lab, we also introduced things that measure like body composition and energy expenditure, because we see that, well, I'm sort of giving everything away now, but we see that the 15q children are smaller, but also lower in weight, and in the contrary, the Angelman children are also smaller, but they're a lot higher in weight. So there's a difference, and UBA3A is known to be involved in hormone nuclear regulation. So that's very interesting to dive in deeper, and that can also be an endpoint in future trials. Yeah. One last thing, just a question. I don't know if I'm sort of ahead of discussing a lot of things we're going to discuss later on, but you say you have also an Angelman clinic. Are there more people here that are involved in both syndromes? No. Okay. Because yeah, I know that. Yeah, because we're actually the last 2 years already organizing a European network for Angelman professionals. So it would be so convenient, efficient if we could combine this. It would cost a lot of time to have 2 networks running, but we will see. And the other thing is the European Registry. I think that's really nice, but the problem in Europe is that you have this privacy legislation, so you cannot put data from one country in a database of another country. So I think the most important thing is that everybody collects their data in the same way. So you can send people syntaxes if there's a question from the network and everybody's okay with that. And you can do your own analysis and then we merge everything. And the last thing, are there centers here that have a PhD that is very active in this field? Because these are the people who are very smart and IT capable. So we can sort of look to have collaborations on that front too, because these are the young people who can bring us further.
So maybe.
Just to the point of the endpoints, we had the metabolic endpoints and some clinical endpoints. We are looking at some genetic endpoints by doing ultra-long read that I guess I'm meant to pass on to Sebastian to talk about a little bit later. So, I'm happy to see that there's all these different areas where we're working towards the same goal from different directions.
Okay, so I think we can all agree that it's an urgent need to somehow harmonize the data, and this will also allow to have more power also for some variables with smaller effect sizes. So that's a really urgent topic.